Hello, everyone, and welcome to today's presentation titled Monitoring and Evaluating CAR T Cells in Patient Cohorts. This webinar is a part of the second annual event in the Cell and Gene Therapy virtual event series. I'm Melanie Chen of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Raquel Munoz Garcia, immunologist at VN del Rosio University Hospital. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Raquel, you may now begin your presentation.
Hello, my name is Raquel Munoz, and I'm immunologist at Hospital Universitario Virgen del Rocío in Sevilla. And I'm going to talk about CAR T-cell monitoring in clinical phase using digital PCR technologies. This is the script that I will follow during the presentation. First, I'm going to explain what the immunotherapy for cancer is, and I will focus on CAR T therapy, digital PCR, and how to monitoring CAR T therapy with digital PCR. First, the immunotherapy for cancer is based on the fact that the immune system has an important role in the development of cancer. The process of immunoediting or immunosurveillance is also called the 3S: elimination, equilibrium, and escape. And based on this important role of the immune system in cancer, many immunotherapy strategies for cancer have been developed, like immunocheckpoint inhibitors, classical T-cell transfer therapies, monoclonal antibodies, vaccines, immunomodulators, but I will focus on chimeric antigen receptors, CAR T-cell therapy. And what's the CAR T therapy is? Well, it's a therapy based on the capacity of chimeric antigen receptor T cells of bind a specific target of the cancer cells and kill them. The process consists on remove blood from a patient and isolates T cells. The rest of the blood is returned to the patients, and in the lab we make the CAR T receptor, and we insert this gene into the T cells and grow millions of these CAR T cells. Then we infuse CAR T cells into the patients, so these cells can find and bind to his specific antigen and kill the tumor cells. This is the schematic representation of a CAR T cell. There are three main domains: the extracellular domain, that is an antigen recognition domain that derives from an antibody and recognize specific targets and determine CAR T immunogenicity. The second domain is the transmembrane domain that is an spacer that provides stability and anchoring the CAR T receptor to the T cell. And finally, the intracellular domain that is a signaling domain that facilitates T cell activation and provides persistence and survival of CAR T cells. This structure have been changed over the time from first generation to fifth generation, And the main difference between these two generations are in the signal line domain. These domains have been improved by adding co-stimulators domains, and in its simplest version, it's also had the CD3 change of a TCR, and it has been adding other co-stimulators molecules. In the last figure, we can see the action mechanism of the CAR-T receptor. The extracellular domain can recognize specific antigens of the tumor cells, and the activation signal go through the intracellular domain and activates the CAR-T cell that kill the tumor cell in a specific way. These are the 6 CAR-T cell therapies that have been approved by the Food and Drug Administration until 2016. And all these therapies have been approved for the treatment of blood cancers, like lymphomas, sometimes of leukemia, and more recently, multiple myeloma. This therapy can lead very good results in some patients, but like all therapies, it can also have second unwanted effects like inflammation, anaphylaxis, and more severe neurotoxicity or CRS, cytokine release syndrome. And in some cases, it can be severe or even fatal. So, CAR-T therapy have demonstrated remarkable clinical results, but also failures and serious side effects. And what it depends on? To answer this question, we need some biomarkers, and peak expansion and persistence of CAR-T are correlated with responses rates and severity of side effects. So, in vivo CAR-T cell expansion following have been recognized as an important surrogate marker for response and disease control. But no standardized method or commercial assay for CAR-T measurement is established yet. For this reason, flow cytometry or quantitative real-time PCR have been proposed to monitoring CAR-T therapy, but both techniques have many disadvantages that no facilitate their introduction in the clinical practice. So we thought, and what about digital PCR? So in digital PCR, the key is the partition. And in real-time PCR, the accumulation of amplification product is measured as the reaction progresses after each cycle. So there is competition of the reagents, and in cases of low-frequency target, it is possible to lose the amplification and only amplify the high-frequency target. But in digital PCR, thanks to partition, we isolate DNA molecules in each compartment. So the detection limit is the same as the quantitative real-time PCR, but the reproducibility is higher, and this is a great advantage for low-frequency targets. One of the great disadvantages of the old platforms of digital PCR was that the significant data volume until a health, limited or inconsistent compartmentalization, a tedious workflow, different devices were used, and a longer waiting time for results until 9 hours. But with the new applied biosystem Quant Studio Absolute Q Digital PCR, the data volume is less than 5%, we had about 20,000 microreaction, and the workflow is super easy. And we obtained the results very fast, in an hour and a half. So we thought about monitoring CAR T therapy by digital PCR. We use samples of 46 patients with relapsed or refractory acute lymphoblastic leukemia or lymphoma B, treated with CD19 CAR T, tisa-genlecleucel, or axicabtagen-thiroleucel. And we use samples in different time points after infusion of the CAR T, days 5, 7, 11, 14, 20 or 23, and 28 and 30. And we obtain genomic DNA from peripheral blood mononuclear cells of each sample. The experimental design consists in the simultaneous amplification of two targets. In the one hand, the FMC63 fragment present in both CAR Ts, and on the other hand, the amplification of RNase P present in all cells. So we used specific primers and a proof labeled with fluorophore to amplify the FMC63 present only in the CAR T construction. And we use a specific set, primer plus proof, labeled with big fluorophore to amplify RNase P present, as I said before, in the CAR T, but also in the other cells. So the workflow is isolate DNA samples, prepare the PCR reaction, load the MAP16 plate, run the absolute queue, and then analyze data. This is a real example of a patient. Here we have the figure that gives you the software. Here we can see the big fluorescence that shows the amplification of the RNase P gene. And here we have the fan fluorescence that shows the amplification of CAR T gene. And the software gives you the copies per microliter of CAR T and RNase P in each sample in day 5, 7, 11, 14, 22, and 30. And to normalize and compare this data between the different days, we applied this mathematical formula and obtained the CAR T copies per cell. As I said before, we know that there are 2 copies of RNase P per cell, so we can calculate the number of cells that it will be equal of copies of RNase P between 2. So here we have the same data but normalized and expressed as the copies of CAR T per cell. And considering all data of the patients, we can say that patients show expansion of the infused product followed by a rapid loss until day 20, after which detection is very low. And if we represent this data over the time in a graphic, we can distinguish 3 groups. Group 1 is the group whose expansion of the infused product occurs between days 10 and 11, and the great majority of patients are in this group, the 82% of patients. This is the typical expansion curve of this group. Group 2, whose expansion of the infused product occurred after day 11, there are only 4% of the patients in this group, and here we have the typical curve of the expansion of this group. And finally, the third group that presents no expansion of the infused product. Also, the 4% of the patients are in this group. So with these results, we can also say that are qualitatively similar to those obtained by flow cytometry with the same samples. And all of these results have been published in Frontiers of Immunology, and this work was made by the hematology group in my hospital, and we collaborated with our data. And at this point, I wanted to thanks, especially to the immunology group of my hospital, for giving me the opportunity. So what about the future? After 20 days post-infusion, the detection of CAR-T is very low, and we have some questions about this. Could digital PCR detect small amounts of CAR-T when flow cytometry cannot? Or could digital PCR be a tool with sufficient sensitivity to detect CAR-T after 20 post-infusion? So we are working to answer these questions. In summary, first, monitoring of CAR-T cells in vivo might be a fundamental tool for the identification of biomarkers associated with response or toxicity at very early time points post-infusion, which will serve to provide earlier interventions. Second, monitoring by Applied Biosystem Quant Studio Absolute Q Digital PCR. Most of the patients show expansion of the infused product between days 10 and 11, followed by a rapid loss until day 20, after which detection is very low. These results are qualitatively the same as those obtained by flow cytometry. And finally, due to its simplicity and reproducibility, digital PCR could be useful in the quantification of CAR-T in follow-up and monitoring. So that's all, and thanks for your attention.
Thank you again, Raquel Munoz Garcia, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd like to also thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Please visit Thermo Fisher Scientific's booth in the exhibit hall after the webinar for more information and resources. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until October 15th, 2026. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.